Welcome to Science Hub. Today, we're diving into the intriguing world of prenatal exposure to pesticides and its impact on brain development. Recent research has revealed concerning effects on the brain due to prenatal exposure to a pesticide called Chlorpyrifos. This groundbreaking study was conducted by teams from Columbia University and other medical institutions and published in JAMA Neurology. The study tracked 270 participants from the Columbia Center for Children's Environmental Health birth cohort. All participants were born to Latina and African American mothers in New York City. Detectable levels of chlorpyrifos were found in the umbilical cord blood of each child at birth. These children underwent brain imaging and behavioral assessments between the ages of 6 and 14. The findings showed a clear pattern. Children with higher prenatal exposure to chlorpyrifos exhibited more pronounced structural and functional differences in the brain. They also performed worse in tests measuring motor speed and coordination. The evidence suggests that prenatal exposure to chlorpyrifos disrupts brain structure, function, and metabolism directly in proportion to the level of exposure. For the study group, household pesticide use was the primary source of exposure. Although the United States Environmental Protection Agency banned residential use of chlorpyrifos in 2001, it remains in use in agriculture for non-organic produce and grains. This ongoing use means that agricultural workers and nearby communities may be exposed to contamination through air and dust. Drive. Virginia Raw, the study's lead author, emphasized the importance of continuing to monitor exposure levels in potentially at-risk populations, especially pregnant women and agricultural communities, where infants remain at risk. The researchers noted that the disruptions in brain tissue and metabolism linked to prenatal exposure to this pesticide were widespread throughout the brain. Drive. Bradley Peterson pointed out that other organophosphate pesticides might produce similar effects, highlighting the need for caution to reduce exposures during pregnancy and early childhood when brain development is rapid and particularly vulnerable to these toxic chemicals. This study underscores the serious and long-lasting impacts of prenatal exposure to chlorpyrifos on brain development and function. With the continued agricultural use of this pesticide, agricultural communities, particularly pregnant women and infants, remain at risk. The findings highlight the urgent need for ongoing research and the development of preventive policies to mitigate the risks posed by exposure to these chemicals during crucial stages of human development. Remember to subscribe to Science Hub for more insightful science content.